Okay, thank you. I also want to thank Patty and the organizers for the invitation to participate. One of my favorite favorite of all times topics to discuss. So I'm super excited to get feedback and discussion and to be part of such an amazing panel. Some of my dear friends here today as well as, and I want to highlight that I know that we have a lot of trainees in the room, especially some undergrads that are also in the audience who have read papers from all of the speakers. So I expect really good questions today. Okay, so I am starting off this prestigious panel and talking about the importance of sex differences, especially as it relates to brain health, and thinking about the developing brain. I'm actually going to challenge you in the next 32 and a half-ish minutes to think about brain development from multiple levels, and that includes even from prior to conception. And so in thinking about how the brain that is housed in your skull today came to be, all the influences that occurred even through development, but prior to that, as a neuroscientist, which many of us in this room are, we have to begin thinking outside of the brain. And that is difficult oftentimes for neuroscientists. Having just come from the Society for Neuroscience meeting, where oftentimes we think about the brain as only being influenced by anything interesting from the neck up. And that oftentimes we forget that the influences of the brain, whether it be brain function, even as you are sitting here today, or all the things that determined the development of your brain began way before that point. So understanding the importance of, as it relates to maternal health, we're very interested in today, and reproductive outcomes, as well as paternal health that I don't have time to talk about today. And so even though I am a neuroscientist, I now tout myself, although I don't put it on my card from Maryland, that I'm a neuroendo, reproplacento, germ cell, androgynologist. And that everything that encompasses the breadth of all the influences on how the brain came to be are really important that we understand as well. This is actually an excerpt taken from the New York Times from December of last year, and it's befitting as our next speaker and I were both in this article, but this particular excerpt is actually from Dr. Weinberger's mention in this New York Times article, which is how Danny and I really began talking about this idea that it turns out that the placenta, surprise, is actually very important for the functioning of the brain. And I'm going to go through some of our data from the last few years, as this model we've been developing over the last decade has really come to understand the sex specificity. And if you look at this article, and I'm sure you'll hear in Danny's talk, that trying to understand not just the role of the placenta as being an important endocrine tissue for brain development, but it turns out the sex specificity of the placenta matters for the brain, as was pointed out from Danny's studies that he'll tell you about, that the male fetuses were more effective in terms of placental relevance to schizophrenia risk than the females. And I think, especially from a neuropsychiatric perspective, as you'll hear again in Danny's talk, that's amazingly exciting to think about the tissues that we have access to and understanding that these genes that we've thought were so important for brain function may also point to periods or windows in development that are critical for disease risk. This is actually from a review that Neil Epperson and I wrote several years ago, talking about this concept of sex differences across the lifespan. And this was around the time that NIH, as was mentioned a minute ago, put out this call for the fact that now submitting a grant that you all know at NIH, regardless of the type of grant, that you have to think about sex as a biological variable in your grant. And the importance of that, this was 2015 when they started talking about this, and how understanding sex differences as it relates to when neuropsychiatric disease presentation occurs tells you a lot about not just who's at risk, but may tell you a lot about mechanisms that are really important for that risk. And I think that this highlights really well, especially on the neurodevelopmental end that I'm going to talk about, which is that we know a lot about neurodevelopmental disease risk being largely impactful for males more than females, and that can tell you a lot about the mechanisms contributing. So I want to start with just a little bit about one of the models we've been working on for the last several years of preconception stress. So we have an NIH grant on both female preconception stress and male preconception stress. Actually, the male preconception stress grant is actually funded by NIMH, and I don't have time to talk about that today, but I spend a lot of time in my lab talking about sperm. And we can, if you come to the reception afterwards and want to talk about sperm, let me know. So for female preconception stress, thinking about how mom's adversity, so everyone's heard of adverse childhood events, that the experiences that you've had across your lifespan up until even the point where you have children can be critically important for determining many aspects of vulnerability for you, but also for your developing fetus and your offspring. So we know that adverse childhood events, which have been well defined, which are outlined here on the left, are important for neurobehavioral and neuropsychiatric risk. And we know from these studies that 4 or more adverse childhood events can be predictive, strongly predictive of disease risk, especially for neuropsychiatric disease. Well, in trying to understand this and model this, it's important, I think, to try and understand what your audience is, what you're trying to model in your aspect, whether it be studying human populations or animal models that I'll tell you about. So across the United States, if you take pregnant women women who are currently pregnant, and look at the adverse childhood events, the number of adverse childhood events for those pregnant women, so either being 0, 1, 2, 3, or 4 or more as that sort of break point, you can see here on the left that across the United States, for those pregnant women, on average, those women that have 4 or more ACEs and who are now pregnant, knowing that that is a risk, again, for them and for the baby, there's about 12.5% of women that have 4 or more ACEs. I live in Baltimore. Baltimore is an exceptionally special population that's dealing with a lot of adversity in its environment. We have a very high African-American population in the inner city as well, and it's something that we're particularly interested in the lab. So if you take a survey of women coming into the University of Maryland Hospital, largely African-American, and look at the number of adverse childhood events experienced by those women in pregnancy, you'll note that it's a lot higher. It's 61% of the women coming in have 4 or more adverse childhood events. And so to understand the impact of the trauma and stresses that they have endured and the impact and risk for their pregnancy and their offspring is very important for modeling. And you don't need to look very far to find all the epidemiological data that points to what those risks are. In fact, we just had a talk at the Society for Neuroscience that has partnered our mouse model with human studies looking at adverse childhood events, especially, and the trauma experienced by African-American women, in order to identify the underlying biology of the perceived stress of racism. And why it's important? Because you can see right here the differences for African-American women in pregnancy for morbidity and mortality is significantly higher in this country, which is incredibly important. We understand a lot of the epidemiology. What we don't have is a lot of the biology behind it. So we're here to talk about sex differences as well. So the battle of the sexes, we largely know from some of the work you're going to hear about from my colleagues today, in addition to that, which is being done by many of you in this room, that neurodevelopmental disorders, the risk for those has a big sex difference. There's a huge bias largely toward males. Why is that? What is it about the intrauterine environment that gives a particular risk for males or possibly or a resilience for females? What is it about the intrauterine environment? Well, if you want to understand the mood of how you model this in rodents, so as Halloween is tomorrow, understanding mood recognition in a pumpkin is probably easier than trying to understand mood recognition in a rodent. It is very complicated, and as many of you probably know, there is a lot of controversy around how do we understand or model neuropsychiatric disease in rodents. It's very difficult. But one of the things, well, with one exception, right, we all know one exception of whose mood is very easy to pick out. But in the average rodent in the vivarium, trying to understand mood is a very difficult, almost impossible thing. But we can model stress. We don't call it trauma because the mouse doesn't understand that it's traumatic or not. But we can model stress in rodents, mice or rats. Mice are largely used because of the transgenics that I'll show you. And we can begin to understand particular windows of vulnerability when chronic stress exposure has happened, because we can limit the experiences that the rodents have in vivarium, obviously. And we can do preconception stress. We can do pregnancy, which I'll talk about. You can do post-pubertal stress, et cetera. You can look at these different windows of development and begin to understand the biology behind the risk. So just a little snippet I'm going to tell you about in studies that have been going on in the lab for the last couple of years. We've begun trying to model the perceived stress of racism. Obviously, you can't model racism in a mouse, but you can, like I said, model stress. And these initial studies began by offering rodents experiences of stress, imparting stress upon them, either males or females modeled here appropriately as pink or blue, and then pairing those animals as preconception stress experience with control matings of the opposite sex, and then looking at the offspring outcomes. And one of the first things we wanted to ask in these studies of either a preconception stressed female or a preconception stressed male was if we just asked if dad was stressed or mom was stressed or both were stressed, and we just used transcription as an outcome. But again, looking at the developing brain of the offspring or the developing placenta, as we'll talk more about, can we start to see differences in the sex of the parent contributing and the sex of the offspring that are the recipients of the preconception stress? So I just want to show you two quick data points before I go back into the pregnancy period. So in these studies, what I'm showing you here is either maternal experience preconception stress, paternal experience preconception stress, or both mom and dad were stressed, again, prior to pregnancy. So this is the maternal stressed, dad was a control, paternal stress, mom was a control, or both parents were stressed prior to conception. What I'm showing you here is data from the placenta from an embryonic day 12.5. That's about mid-gestation in a mouse. And what these colors are showing you for all gene sets, this is a total transcription RNA sequencing comparison of the placenta taken at mid-gestation, one male, one female placenta taken out. These are for the female placenta, showing you there are lots and lots of differences. Blue indicates an increase in gene expression. What I'm showing you here is the size of the circle indicates the number of genes in that gene set. The bigger the circle, the darker the color, the more the changes, basically. And what you can see that if you look at those changes in both parents being stressed, who's really driving those changes in the female placenta is dad, right? So largely they're lining up around metabolism and things relative to transplacental signals that would give cues to the developing fetus about nutrition and the environment. But again, dad's stress experience is driving the female placental changes. What's really intriguing, again, related to this male risk during in utero experiences, you see something very different here for the male placenta taken. Again, if mom has been stressed, if dad has been stressed, or if both. And now what you see is something very different. The majority of genes here, which are shown in red, are actually decreased in expression. There's a lot more of them, and mom's stress experience now is driving the male placenta. So the female placenta from the uterus was being driven by dad. The male placenta in the same uterus is being driven by mom. And I think that's incredibly interesting. And if you begin to look at what these gene sets are, there are a lot of them related to immune function. So not only do you have a sex difference in the contribution of mom or dad, you have an incredible sex difference for the recipient in the same uterus being a male or female placenta. So because I'm the first speaker, I'm going to highlight again, just for those who don't work in rodent models, that because the rodent obviously has a litter, not just one, it's not a singleton birth, right? It's a litter. You have males and females gestating in the same intrauterine environment. And yes, every developing fetus, just like in the human, has its own placenta. I like to point that out because sometimes people have this vision, I don't know why, of the rodent being this gigantic placenta and all the fetuses hanging off that. Every developing fetus has its own placenta. The sex of the placenta, obviously, is the same as the sex of the fetus, and we'll come back to that. So the take-home message here, I think, is incredibly interesting. Not only is it sex of mom or dad and their environment that matters, but sex of the developing fetus itself is obviously important. So now I'm going to return back to studies we've been doing over the last decade or more, really asking about these sex-specific mechanisms of the placenta. Since we know that neurodevelopmental disorders have a much higher rate in males than females, we know that intrauterine environment, all you need to do is talk to a NICU nurse or a pediatrician who will tell you that girls go home before boys, there's more boys than girls in the NICU. There's something about that intrauterine environment that is either, again, a risk factor for males or a resilience factor for females, either or. So what is it? So we began thinking about these studies in terms of a model in our lab that we developed well over a decade ago where we can take mice, we can stress them starting in pregnancy. Initial studies did early, mid, and late gestation. I'm just going to summarize here many decade or more of work, which really summarized as early gestational stress. What's happening in early gestation in mouse is the equivalent really of the first trimester in humans. And in these studies, it is the male, not the female offspring that show a phenotype. The females don't show any phenotype at all. If mom is stressed for the first 7 days of gestation, it's a chronic variable stress. We're just doing something different to the pregnant female every day. Mice are very reactive creatures. They react to everything. They habituate almost to nothing. And so it's very easy to stress them. And so in this model, when these animals are born and when they're adults, the male offspring, not the female offspring, show a sort of a basket of neuroendocrine phenotype, meaning they are more stress reactive. This just shows you an example over here. This is corticosterone on the y-axis. This is time. This is an adult. These are adult. This happens to be the male showing you the phenotype. When they are adults, if you give them an acute stressor and measure their stress hormone change, you can see that compared to controls, those early prenatally stressed male offspring are now showing an exaggerated stress response. I like showing the HPA because it is all the same players and all the same axis in humans as it is in the mouse, right? So it's very translational. So that's changed. The stress regulatory genes are changed in the brain as you would expect. They show some cognitive deficits, which I don't have time to talk about today. But around this neuroendocrine basket of phenotype, these animals also show this really interesting difference. Again, males, not females, where they're born totally normal, and up until weaning, these animals have a normal growth trajectory. Once they're weaned, which I will point out for the endocrinologist in the room, Ursula, that when they are weaned, coincident with puberty, they also drop down about 10 to 15% in body weight and growth. This is not a model of a disease. This is not autism or schizophrenia. What this is showing you that very mild stresses in a very specific window of development produce lasting changes in the brain, specifically in males and not in females, that may underlie an added risk. So now if you took, as Danny will talk to you about schizophrenia from the GWAS, for instance, you took gene risk, this might be the environmental trigger that then puts somebody at risk for things like for any neurodevelopmental disorder. Okay, so males show the phenotype, females do not. So we began asking the question then molecularly, what is it about the placenta, and I'll show you in a minute why we started thinking about the placenta, that may posit this risk? Why in the same uterus with the same experiences are males showing something that the females are not? So we started by looking at the placenta. This was years ago. This is from a microarray, where we began asking what statistically robust sex differences exist in the placenta from a gene expression viewpoint, meaning your hypothesis could be a case in which males and females start off at the same baseline in terms of placental responses, right, to the environment. They start off at the same point, but they respond differently to a perturbation in the environment. And that perturbation could be immune, nutritional, stress like we're doing here, right? So they're responding differently, or it could be that they start off at different points and respond the same, but because of the starting point of one of them, they're at more risk. So we wanted to know, what are the differences in a baseline way of looking at male and female placenta? This is looking at embryonic day mid-gestation, 12.5, 15.5, and 18.5, which is about a day before birth. And what we found was, as you would expect, there was not a lot of genes that met this criterion of being robustly, significantly different. But the genes that were all either X or Y linked, meaning they're likely escaping X activation, so they're twice as high in a female placenta, right? We're talking about the trophoblast cells of the placenta, largely here. So they're either escaping X activation or they're on the Y chromosome, and that's what all of these 7 genes are. So there's not a lot of differences, but they're really interesting when you start to look at what their functions are. And why is that interesting? Because if you don't study transcription, you may not think this way, that if you are an enzyme or a factor, transcription factor or an enzymatic process that is significantly different, it positions you in a way to respond to an environment during that perturbation to produce long-lasting changes, right? So you can have methylation changes, you could have enzymatic changes in histone processes that allow a female or a male placenta to therefore respond differently to a change in the environment. Okay, so just for time's sake, I'm only going to tell you quickly about one story related to one of these genes, OGT, or O-glycosyltransferase. It is an enzyme that is X-linked. Therefore, it is expressed only from the X chromosome. So the levels, it seems to escape X inactivation. The levels are twice as high in a female placenta than a male placenta. And that includes human as well as mouse tissue. We've done both. This enzyme, because it's escaping X inactivation, starts off twice as high, like I said, in a female placenta than a male placenta. OGT turns out to be an incredibly interesting enzyme. One of the reasons is that, so evolutionarily, if you're going to pinpoint something to have a functional role to be important for what may be neurodevelopmental disease risk, this enzyme is tightly regulated by glucose in the environment. So if you are interested in gestational diabetes, for example, maternal obesity as another example, those factors contribute largely to regulation of this enzyme. This enzyme is also critically important at the level of the cell for what it does. So this enzyme is really important for epigenetic regulation, which I'll show you in a second, but also for its loci. So it turns out OGT loci on the X chromosome is located really close to the long non-coding exist, We know that exist obviously is critical for exon activation, and recent studies from other labs, not mine, have demonstrated that there's an interaction between the oglycanacylation mark that responsible from OGT and exist regulation, meaning it is possible that in different environmental or developmental time courses, there may be a biochemical way for risk or resilience for the female brain, depending upon the interaction between these two processes of where genes are exon activated or maybe not exon activated. And so we'll come back to that. Okay, just quickly, this is the biochemical readout from our differences in our placentas. This just shows you the mRNA, just like this is the protein here, twice as high. And even more importantly than its mRNA or protein levels is its function. So OGT is an enzyme. It places an oglycanacylation mark. Say that twice fast. And a glycylation mark on serine-threonine residue. So oftentimes you hear about residues in proteins that are phosphorylation events, typically phosphorylation being an active or a GO, right? That's the gas. Oglycanacylation of hundreds and hundreds of proteins tends to be the break. So they sort of work in tandem to go or break depending on the system and the cell type. And so because the levels are higher in a female placenta, so is the oglycanacylation marks from total placental proteins, right? So we know that functionally, the level, the titration of this enzyme is important for the amount of oglycanacylation mark, and therefore may be a readout in terms of function of what all of those proteins are doing to respond to an environment. In this case, this is our sort of going hypothesis here. These are the levels of OGT in a female versus a male placenta with stress. So they're starting off at different levels, twice as high in a female compared to a male. Following maternal stress at mid-gestation and the remainder of pregnancy, the levels drop very low, but much lower in the male placenta than the female. Therefore, perhaps a threshold of vulnerability whereby the level of this one protein and its activity may fall below where now there's more GO or more gas on those proteins than there would otherwise have been, putting the transplacental signals being relayed to the developing fetus to be different in a male and a female. Okay, so why doesn't the female placenta respond to those intrauterine perturbations at the level of the cell? So now I'm going to ask you to go from mom to the fetus to the placenta all the way down to the trophoblast cell of the placenta. And I'll take you back to your days of developmental biology where you learned about reproduction and post-fertilization development, right? You had the zygote, the embryo, 2-cell, 4-cell, all the way up to the blastocyst. The blastocyst, which obviously is either XX or XY, inner cell mass will develop into the fetus. The extraembryonic tissue or the trophoblast, the trophectoderm of that blastocyst becomes the placenta. And those are the trophoblast cells. By embryonic day 12.5, which is why you often see that in my talk, that is the mid-gestation point, but it's also the point where all of those trophoblast cells that differentiate to all the required cell types of the placenta in a mouse have fully differentiated, right? So we do our stress days 1 through 7. This is now 5 days after stress has ended. We're looking at these differences. Okay, so what does OGT do and why do we care? Down in the trophoblast cell of the placenta, OGT does a lot of things. Like I said, it regulates hundreds of proteins. One of the key things it does related to epigenetics and transcriptional regulation is it stabilizes the methyltransferase EZH2. EZH2 is a methyltransferase specific for histone 3 at lysine 27. And for anybody who remembers their transcriptional days of studying gene transcription, you remember that histone 3 lysine 27, when it's trimethylated, H3 K27 trimethylation is a ubiquitous transcriptional break. It's a transcriptional repressor, meaning the more of this you have, female, the more of this you have, female, the more break you have, female. And thereby, at the level of the trophoblast cell in the placenta, you have all the way down at the level of the transcriptional control, mom's diet changes, mom's immune changes, mom's stress. You have a way in which females probably can place a greater brake on transcription that males don't have. So in fact, if you look at the levels of this repressive mark, you can see again in female placentas compared to males, you have way more H3K27 trimethylation. I often get asked why, and so I'm just going to point out here, the probable why is, possibly, is that this mark is also required as part of exon activation. So it may be in this extraembryonic tissue a byproduct of exon activation in these trophoblast cells that allows females to more tight control their response during development than males, at least specific and perturbations. Okay, so just quickly, because we're here as neuroscientists and wanting to understand the brain, the trophoblast cell type specificity is one of the things that has been difficult over the years to wrap our heads around. When we take out a placenta, be it human or in this case mouse, you get many different cell types, right? The placenta is highly vascularized. You have maternal decidua in there as well, which of course is going to be XX. And we always worried that this may have confounded some of our outcomes. If the transcriptional control in these cells is so robust, you'll recall that I only showed you there were 7 genes that were robustly sex different between a male and female placenta at baseline. And that may be because it just poises that cell to respond differently, but it may also be because we're getting contamination from all those other cell types. So we decided we wanted to ask, in a very molecular way, could we get at just the trophoblast cells of the placenta, meaning just the XX and XY coming from the fetus, to ask about these differences in development. So we used, and one of the main reasons we use mice in the lab, because of transgenics. So this is called ribotag. It's a transgenic mouse that allows you in a conditional way, so driven by a specific promoter, in this case, a placental-specific promoter. It allows us to drive expression of an HA or a hemagglutinin tag placed on only in specific cell types on a subunit of the ribosome. And what that allows us to do then is to pull down from, you could take the whole mouse and grind it up, but you're only going to get ribosomes taken out of trophoblast cells of the placenta, and that allows you then to get rapidly translating RNAs and cell type specific RNAs. Okay, so when we did that, so what I'm going to show you here is sex differences by RNA sequencing now. So you remember before, we only found 7. We are now doing RNA sequencing, so of course, now you have greater sensitivity, so that accounts for some of these differences. But what I'm showing you here is RNA sequencing comparing a male and female placenta at that same time point. And so we're looking at log 2-fold change on the Y chromosome here, and mean normalized counts, meaning higher expressed genes and lower expressed genes here. A red dot indicates a statistically significant difference between a male and female control placenta, meaning when we use just trophoblast cells as our outcome measure, we see now a lot of sex differences. Okay, that's great. But what about OGT and its role? So now if you take a placenta in the same sort of format, from a female where we've knocked down the level of OGT using the same Cree driver. So now we have hemizygous OGT female should be at the same level as the male, right? Because male is XY. So we've knocked down. So now we have the same level of OGT in the trophoblast cells of the placenta. Now compare the sex differences in the placenta, and they almost all go away. OGT seems to be the driver in the trophoblast cell of many of those sex differences, which is really intriguing. In fact, the only ones that remain are genes that are escaping X inactivation or those that are on the Y chromosome. Okay, so what does that have to do with the brain? Many of you in this room are very familiar with the developing brain being sexually dimorphic, especially the developing hypothalamus. That's critically important. Here I'm showing you RNA sequencing heat map from a normal, this is showing the genotype of the placenta. This is the brain, though, from that fetus. This is a normal XX. So OGT levels are twice as high, right? Placenta, this is the brain. This is a normal OGT placenta male. You can see vast sex differences in the gene expression pattern, as you would predict, knowing that, of course, as the testes are developing, they're producing testosterone, right? Testosterone from the testes enters the brain, is aromatized to estradiol, kicks off that event of the sexually dimorphic brain. We've all heard that a million times. So we wanted to know what role OGT in the placenta may play in the hypothalamus of this developing brain. So again, from that placenta I just showed you where we knocked down OGT just in the female placenta, so its levels are the same as the male. And now, whose brain does it look like? Well, it turns out it looks a lot more like the male's brain. So something about the transplacental signal that is being produced from the placenta interacting with and programming the developing brain is critically important for those sex differences, at least within the hypothalamus. Okay, so just quickly, because I'm sure we're running out of time. We also wanted to begin to understand in this model, right? I've now shown you some of these differences we know are important for the brain as it develops, important for outcomes and behaviors, important for stress reactivity, important for sex differences. The question still is, after a decade or more of working in this, is what is the transplacental signal? If we think, or at least we hypothesize, that OGT and what it's regulating in the trophoblast cell is important, we still haven't identified what the transplacental signal is. So I'm going to show you some outcomes. We began by doing this also in collaboration with Alex Bonin, who is at USC, did a lot of his initial work with Pat Levitt, thinking about the role of serotonin production from the placenta. And due to that work, he established, and as far as I know, is one of the only people in the world, much less in this country, that can do this amazing process where he can take out the mouse placenta during gestation, hook it up and keep it alive and happy, perfuse on one side and collect the perfusate off the other side. And that is really the only way to really get your head wrapped around what is being produced by the placenta that will be making to the developing fetus. Okay, so what are we thinking about here? Well, in thinking about how the signals, the transplacental signals being produced by this placenta or this sex-specific endocrine tissue, we're thinking about it in multiple ways. We're approaching it from, based on the data I just showed you, are there steroid hormones being produced in a sex-specific manner that are influencing brain development? Are there extracellular vesicles we know are produced in large part from the placenta that may be interacting with fetal development? I'll talk about those. And then, of course, are there just other proteins that we haven't thought about yet? So just quickly, because I know this is of interest to Jill Goldstein, who's sitting here, from some of her data, that one of the things that we did early on in these studies when we identified OGT is to take that O-glycanalkylation mark and use it to sort of feed into our hypothesis about what the targets might be in the placenta to give us a clue about what to look for in the developing brain. When we did a chromatin immunoprecipitation sequencing, or ChIP-seq experiment, with a glycanalkylation mark and overlaid it with permissive and repressive marks, only one gene candidate came from that, and that was 17-beta HSD3, which is super interesting for the endocrinologists in the room, because this one particular enzyme is important for the conversion of androstenedione into testosterone. And at the time, we were intrigued by this finding because we know that our phenotype and our early prenatal stressed male offspring always puts them sort of in a female-like direction. So any assay where we see a sex difference, the males and their behavioral responses, their stress reactivity, their brain differences that I just showed you, kind of looks like more in a female direction. So could this be something that is coming from the placenta that's important? So we know, but looking back at levels of this enzyme in the placenta, the male placenta, it is dramatically reduced. So that would make sense. As well as when you look at the testosterone levels of the placenta at embryonic day 12.5, it is significantly reduced by stress days 1 through 7, as well as the androstenedione levels are increased. Why is that important? Well, because if you also look at our animals, their serum testosterone levels as adults are lower. The anogenital distance of these animals is decreased, suggesting a reduction in testosterone exposure, and their testes weights are smaller. Again, they're fertile, they're perfectly normal, but it just suggests that there may be something about masculinization that occurred. Why is this important? As Dr. Goldstein is sitting here, and when I was at Penn, the GERS worked on this as well, and Joe Goldstein's studies also showed the same effect, which is that if you look at a cohort of schizophrenic patients, the male brain, especially early onset, I believe, although Jill can speak to this, that there, first of all, the sex bias in that early onset of schizophrenia presentation associated with fetal antecedents as well, but also low serum testosterone levels, that oftentimes when you look in the brains of these individuals, the male schizophrenic brain in many of the brain regions looks more female-like than male-like, which is not to say that being more female-like is more schizophrenic-like at all, but it is to say that it points to a particular window of development, again, or a vulnerability in utero, where males normally receiving those signals for the sexually dimorphic brain are not receiving them. So that's something that we're currently looking at, again, by looking at these placental perfusates with Alex Bonin. So just to touch upon a little bit of the proteomics piece of this, we began by taking, again, these placental perfusates and asking, can we find any clues there that might suggest support of a sexually dimorphic brain programming? We looked at over 400 proteins that were detected in the perfusates from our male and female placentas, control and stress. 400 proteins that were detected in this perfusate, and of those, 0 of those proteins were detected in the male perfusate that were not also present in the female perfusate. However, there were 7 proteins we detected in the female perfusate that were not at all detected in the males. So there may be resilience factors, possibly, again, with that risk-resilience balance there, those 7 proteins, and they're all kind of looking like they're maybe around something to do with either EV signaling or immune signaling, which is something that we're also following up on. And so extracellular vesicles is a big focus of my lab as well. It's something that we look at related to both within and to the brain, but also in the paternal stress transmission, which I don't have time to talk about today, for how the male or the dad signals to the maturing sperm. For those not familiar with extracellular vesicles, you've probably heard them called exosomes. Extracellular vesicles in mammals are produced by every tissue. They travel around in circulation. They're super exciting, again, for all the endocrinologists in the room. They're super exciting because like an endocrine signal, they have a much greater specificity for their signal. So they travel from a tissue to a tissue. Largely, the cancer biologists and immunologists have figured out a lot about these signaling molecules. There are proteins in the membranes of these things. They're teeny tiny. There's all different kinds of them, which is kind of shown here. There are multivesicular body transmitted. So there's microvesicles, there's exosomes. There's all kinds of signals that are released. Up until about a decade ago, we thought they were all garbage that were being released from tissues and cells, but it turns out they're actually signaling molecules. And unlike endocrine signals like estrogen or testosterone that can travel and act in any tissue, these signals have incredible specificity, and I'll show you in a second what I mean. They have proteins in their membrane that tell you a lot about where they've come from and where they're going, but their content is largely non-coding RNAs. And so when they travel to a specific tissue, and they are largely secreted during pregnancy. There is no other time when you can detect so many in circulation as you can in pregnancy. The placenta produces a ton of them. That they travel in circulation to distant tissues to produce a signal at those cells, largely related to the non-coding RNAs, largely microRNAs, which are incredibly interesting. Okay, so just some quick pieces of data. We've begun asking questions about these extracellular vesicles. So when we stress mom, wanting to understand when you collect in circulation from the female that is pregnant, we started looking at the microRNA content of these. And what you can see based on what I just told you, first of all, we're going to show you the number of extracellular vesicles in circulation is enormous in pregnancy compared to non-pregnant. And this heat map just speaks to that. So when you look at the content of the vesicles, this is just a concert of some of our data and the specification of how we purify the vesicles. Just showing you that the big difference here really in the content is between non-pregnant and pregnant in circulating vesicles, right? Remember, every tissue produces them. And if you look at the pathways that would target from those microRNAs, all really interesting toward brain development. And that's something that we're following up on. This is looking at the protein content that Bridget did last year, which is looking again at non-pregnant. Big differences in pregnancy, of course, as you would expect, but also big differences with comparative stress versus control. Again, a lot around the immune system and a lot of sex differences depending on the fetus that you're collecting the EVs from versus circulation in mom. So a lot about these EVs, I think, are the translational potential, because obviously you can easily collect them from humans. They are abundant in urine, they're abundant in saliva, they're abundant in plasma. So you can collect them from a lot of different tissues and ask in a repeated way that I think a lot of our questions in a translational way you can ask. This just shows you that when we take the extracellular vesicles and collect them from a pregnant female, fluorescently label those extracellular vesicles and inject them back into a pregnant female, which are studies that Katie Morrison Lab is currently doing from control and stress, and ask if they distribute differently. This just answered our initial question was, this is a big question in the field. A lot of placental biologists are very interested in extracellular vesicles. Do they cross from mom into the fetus? The answer that we're coming up with here is a big solid no. So this is showing you when you inject them back into mom, they clearly end up a lot in the placenta, nowhere in any of the fetal tissue that we could find. So they didn't seem to be crossing even 24 hours later. However, they do get into mom's brain. So if you inject them back into mom, they have an incredible specificity getting back into mom's brain. These are coronal sections showing you they're not just on the outside, they're actually entering into the brain. And so for those interested in many aspects of pregnancy and women's health, I think that also is critically important. So I know I covered a lot, but I want to try and finish on time. So in summary, we've really taken this approach of understanding both mom and the developing fetal brain and sex differences therein. I didn't have time to talk about all the data today on preconception male stress data, which I think is also incredibly important. I know a lot of people in this room study perturbations and epidemiological outcomes of mom's stress prior to and during pregnancy. I want to really encourage studies that include dad as well. We have a lot of evidence that the stressors in the environment are carried through extracellular vesicles with dad's sperm at conception, so very important. In these studies I showed you today, the placenta is a very sex-specific tissue all the way down to the level of the trophoblast. We're now currently trying to understand taking those trophoblast cells as they divide and differentiate and look at the functions of those placentas and the transplacental signals as well. We've identified O-glycosyltransferase as a really critical enzyme. We've done studies both in our mouse model and in humans. that demonstrates its sex specificity and that it may be one marker, possibly a biomarker, but also a marker predicting transmission of information from mom, gestational diabetes, preeclampsia, et cetera, all these important factors and perturbations to, in a sex-specific way, the developing brain. And within these trophoblast cells, we think that what OGT is doing is that it's largely regulating this transcriptional responses to perturbations, largely through regulation of H3K27 trimethylation, possibly also through interactions with exist. We're still doing studies on that. And that in this sex-specific manner, many of the outcomes that we're seeing in the offspring brain, the male offspring developing brain, really have a lot to do with understanding the hypothalamus and its development, energy regulation, and mitochondrial function. And those gene sets, which may be predictive of the fact that we're always looking at the hypothalamus in my lab, but I think it is evolutionarily preserved a mechanism by which any perturbation in the environment, the biggest stressor of all, obviously, is food availability, right? So the common stress signals may be relaying information about a stressful environment, programming within the hypothalamus, which we all know that is grand central station, right, for integration of the environment and survival of the species, for growth, circadian rhythm, stress, metabolism, reproduction, right? So it may be why we're seeing these same gene sets. And so just as a final conclusion, I think, and I want to encourage, I hope, whether you're working in animal models or the human, that sex differences at the level of the placenta are vitally important, both for trauma experiences and stresses of mom or dad, but in sex differences in the developing brain. And so with that, I want to do the most important part, which is thanking all of those who've done the incredible work from my lab. Bridget Nugent had really did the most of the work on this placenta and really trying to understand these transcriptional outcomes. And this is also a takeoff from a lot of the studies from Jennifer Chan and Chris Morgan in the lab of doing one of the paternal studies as well. And Katie Morrison, has also been working on a lot of the extracellular vesicle outcomes and looking at the proteomics and transcriptional responses therein, and our incredible collaborators around the country that I've been working with, including Alex Bonin at USC. We did a lot of studies on humans, and as well as the tissue that we got from Neil Epperson's group at Penn when we were both at Penn, and a lot of our epigenetics from the Epigenetics Institute at Penn as well. And I'm happy to take questions. And our funding, of course, our funding for sure, we're absolutely very ecstatic about the funding that we've received across different institutions at NIH. And we do a lot of work on Twitter to advocate for studies related to both racial differences as well as sex and gender differences on Twitter. So please follow us.
Great, thank you. That was terrific. We have time for questions. Who's up first?
I'm curious about the time points of affecting the fetus. So it seems like the sperm only has preconception sperm to influence the fetus, but the maternal influence starts very
Sure.
from the.
Yep.
And can you comment on that time frame and how what role that might play?
That's a really intriguing question. So those are some of the studies that we're doing now and trying to figure out these specific windows. I think the more that we can appreciate the windows in which vulnerability occurs and for which sex can mechanistically point to a lot of things about neurodevelopmental disease risk. So the preconception male studies that we have a whole separate grant and project that works on that have looked a lot at the timing of stress insults, preconception, obviously. In the rodent world, dad does not need to be present, obviously, during gestation or postpartum. In the animal models that we do, dad is only present for one glorious evening, and then he's removed from the cage. And so he is not present during gestation, and he never sees his offspring, and yet imparts some pretty significant effects on the developing brain, not in a sex-specific way as it relates to just dad's stress and the brain, although we do see differences in the placenta. Mom, obviously, is a total different thing. So we have a separate project, which I didn't have time to also talk about today, that's funded by NIEHS, which looks at female preconception stress, which only occurs up until pregnancy. We don't do, in that model, stress during pregnancy. However, as you're pointing out, it's very difficult to remove the effects of preconception stress environment from effects during gestation, even if you're not continuing the stress. So there really isn't, unfortunately, a way in which to continue that unless, and those are studies we're currently working on, doing like embryo transfer. And so you can sort of look at the preconception environment up until a certain point and then do embryo transfer. We do a lot of studies, especially with our preconception models, especially the male. We do ICSI. So you can do intracytoplasmic sperm injection. So we know that vesicles in that model are particularly important for transfer of that information. So we're getting at those time periods by doing those sorts of things. Unfortunately, since we work on stress, one of the difficult things is when you start messing around with surgeries and transfers, you introduce all other kinds of variables, right? Anesthesia exposures, pain from surgery that you can't discount. And so it sort of gets at timing, but it introduces other confounds. But that, to our knowledge, that's one of the better ways to get at that question. But it's an intriguing question.
Next intriguing question. Okay, I have one.
Wait, where's our undergraduates?
Yeah, Tracy, speak up, don't be shy.
Tracy, I may have misunderstood that the exosome data with the stress pregnant mice looked like the immune pathways were depleted in those exosomes. That was a minus Z value.
Yes, so the immune function, which is usually heightened in males compared to females, but within males from the stress groups, the extracellular vesicle targets for those proteomics data, seem to be reducing that immune response. And we've seen that as well in the placenta with our maternal stress studies as well, the sex specificity and the targets therein. So Joel Sadowski at Pittsburgh, at the McGee, has done a lot of work, right? He's really done the most work, I think, around extracellular vesicles during this period of development. And he looks at placental production of extracellular vesicles as they relate to function, again, within the placenta, sort of how they work within as a more of an autocrine sort of function. And again, sees this sort of factor that extracellular vesicles are critically important for signaling to the immune system, which is why a lot of cancer biologists have studied them as well. So you're right. So we see a reduction, and whether those signals from the vesicles are related to a reduction. When we look within these animals, those vesicles uniquely, one of the target tissues uniquely is the thymus. So something about that immune response.
I had a question. That was really great, thank you. I was curious if you could speak at all to whether there was an effect of different types of stress. You mentioned food restriction as one example.
Also a great question. So I've changed my thinking about this over the years, right? So, a decade or more ago, we used to talk about stress for animals as, psychological stress or physical stress. And, when it comes to a mouse, that's all this stress, right? And we do a lot of work at the molecular level. So we're interested in, the mineralocorticoid receptor versus the glucocorticoid receptor, right? Because circadian rhythm in mammals really rises and falls in a 24-hour cycle, really related to your baseline and stress response anyway. So our question is always, well, how does a cell know? How does a trophoblast cell know, oh, this is stress, oh, wait, no, this is circadian, right? And so we look at it sort of as a disruption of normal circadian rhythm, and you're switching from a mineralocorticoid receptor to a largely glucocorticoid receptor function at the cell. When it comes to differences between food deprivation or obesity or immune function or low protein or whatever your perturbation is, the studies we've done in the lab, especially related to placental function and male-specific vulnerability, we've done a maternal immune activation years ago, and we've done maternal high fat. And we always see within the placenta the same thing, where at the same time point, the male placenta, no matter what the perturbation is, shows all kinds of transcriptional responses, and the female placenta really just doesn't. Like I said, it seems to just have this calculated break on it that maybe you have to just push further to see a response. So from the level of the placenta, it doesn't seem to matter what the perturbation is. The outcome and the pathways is very different, but the sex specificity is not. So there was a question, yeah.
So I have two questions. One of them is, do you know when the X inactivation starts? And whether
Great
for
question.
syndromes where you have XXX, is there triple the amount of?
Fabulous questions. Those are really, really great questions. So X inactivation, depending on the cell type. So around that point, there's really interesting study that came out last year from Montserrat Aguirre at Penn that looked at X inactivation or X exist clouds. and she looked within immune cells and from people who have autoimmune diseases and found some really interesting differences around X inactivation, and that in some cell types, that regulation is very different. We've not looked in the placenta. I'm assuming that soon after cell differentiation types are happening, you're getting that X inactivation. The thinking, the dogma has always been that once an X chromosome is inactivated, it remains X inactivated, but there's some recent cool studies coming out that suggest there may be ways, that's why I was pointing out that glycanalylation thing, that seems to be that ways in which prior X inactivation, some genes can re-escape, which, that's so interesting, yeah. And then related to women or men who have multiple, right, so trisomy of XXX or X or OY or, sorry, XY or XO. There's some really, we actually did, Neil and I have in one of our reviews a whole discussion on that aspect. There's some really interesting differences. My take home from that was the number of X's matters more than the number of Y's. So the number of additive X's has an effect, whereas the number of additive Y's doesn't seem to, and that may be obviously for the number of genes expressed off of the X. But I want to point out that while we spent a lot of time as a community thinking about the X chromosome, the Y chromosome is back in fashion, and there's a lot of really interesting things expressed off of the Y chromosome as well. So.
Great. Well,